हेलो एवरीवन। आज का वीडियो लेक्चर है सदर्न ब्लॉटिंग पे। We all know that molecular diagnostic is rapidly becoming a standard laboratory procedure for a large number of disorders. Many DNA-based molecular techniques are being used in clinical practices. In addition to the diagnosis of a particular patient, molecular diagnostic can be used for the diagnosis of index cases, prenatal diagnosis, and screening. In many cases, the diagnosis is based on detecting the mutations in the gene involved. But in some cases, it may be detecting changes in DNA copy number, which leads to change in gene expression. Still further, there may be changes at the transcription level, that is, the production of increased mRNA, which leads to increased protein synthesis by the corresponding gene. In this video, we will explore Southern blotting technique. So, let's start. Blotting का मतलब होता है immobilization of sample onto a solid support. ये sample protein, DNA या RNA कुछ भी हो सकता है. It involves the transfer of nucleic acid or proteins from a gel strip to blotting membrane, that is nitrocellulose or activated nylon. ये nitrocellulose membrane reactive matrix है on which the nucleic acid or proteins become covalently bound in a pattern similar to that present in the original gel. Southern blot technique is based on the specific base pairing property of complementary nucleic acid strand. This technique is therefore based on DNA hybridization. Similarly, in the northern blot used to demonstrate the specific RNA. In this, the total RNA is isolated from the cell, electrophoresed, and then blotted onto a membrane. This is then probed with radioactive complementary DNA. There, RNA-DNA hybridization will occur, and this technique is used to detect the gene expression in a tissue. Now, the third and the last blotting technique is Western blot analysis. It is used for proteins. In this, the proteins are isolated from the tissue and electrophoresis is done. The separated proteins are then transferred onto a nitrocellulose membrane. After the fixation, it is probed with radioactive antibody and autoradiograph. In today's lecture, I would be covering the topic of Southern blotting, and in this we will be learning about principle, steps and importance of Southern blotting in detail. Blotting is done by enabling the mixture of molecules pass through a gel that separate the molecule based on their molecular size. The molecules being tested are hard pressed against a membrane which will then transfer the molecule from the gel onto the suitable membrane through the capillary action. The Southern blotting method is a classical technique that separates the DNA fragment according to their size through electrophoresis. They will be transferred to a membrane, hybridized, washed, and lastly detecting the labeled DNA band. Now the question comes, what does the Southern blotting technique reveals? It reveals or identify the DNA molecule. Secondary, abundance of the DNA molecule and the size of the DNA molecule. So, how much DNA is needed for the Southern blot technique? The amount of DNA needed will vary or will depend on the size and specific activity of the probes used. What is the principle of Southern blotting? Southern blotting is basically a restriction fragment length polymorphism, and it is a hybridization based method for identifying the size of DNA from a mixture of other similar molecules. This technique has high resolution and utilizes the probe sequence for DNA. Basically, Southern blotting separates DNA fragment by gel electrophoresis. The DNA fragments are identified using a labeled probe hybridization. Since The probe is attached to a label such as fluorescent chemical isotope. The bound sequence can thus be visualized. This is the principle of Southern blotting method as well as its variants and require the target nucleic acid sequence to undergo the separation by agarose gel electrophoresis, transferred to an appropriate membrane, mainly nitrocellulose membrane, and then treated with a solution containing the labeled probe that will be fluorescent or colorimetric. DNA fingerprinting has revolutionized the crime investigations across the world. The ability of DNA analysis to prove that a biological sample is a genetic match with the crime scene sample has unleashed the potential of this technology's use in the crime or criminal justice system. Criminals who have committed rape, murder, now face the scientific evidence as well as a human judge. They might have been able to circumvent the evidence in the past, but with DNA fingerprinting, it is almost impossible to escape the law. The innocent people who are accused are also exonerated using this technology. It is due to this reason that the use of DNA evidence in court is accepted in most countries today. Further, the creation of DNA data banks for the criminal investigation has empowered the investigative authorities to get the significant leads in their cases. This has helped tremendously in capturing the guilty, freeing the innocent, and increasing the conviction rates, especially in the sexual crimes. The success of DNA fingerprinting as a crime fighting tool is known to almost everyone these days. Before going into deep, I want to mention you some facts which you should know when we are studying Southern blotting applications in the forensics. Actually, Sir Alec Jeffrey invented the DNA fingerprinting at the University of Leicester in the UK in 1984. It became a worldwide phenomenon when this technology was successfully used in solving the case of the rape and murder of two 15-year-old girls in England in 1987. After creating this technology, Jeffrey sold the patent rights to a private company. It was in 1988 that Dr. Lalji Singh Tandon created his probes through his research on using the snake venom for DNA profiling in India at the Center for Cellular and Molecular Biology, CCMB, Hyderabad. After successfully experimenting with the technology in his laboratory, he offered this revolutionary new technology to be used in a case involving the dispute over the identity of a child by two different couples. Thus, India became the third country in the world after the UK and the US to have the technology for human DNA fingerprinting. Professor P. M. Bhargava encouraged and supported Dr. Lalji Singh in using this technology for providing the forensic DNA evidence in the courtrooms to help in fighting crimes and putting the criminals behind the bars. To clear this concept more, we can say DNA fingerprinting एक laboratory technique है जिसके द्वारा हम establish कर सकते हैं link between the biological evidence जो crime scene से मिला है and a suspect in any criminal investigation. यहाँ DNA sample जो कि crime scene से मिला है उसको हम compare करते हैं DNA sample से obtained from a suspect. If the two DNA profiles are matched, then the evidence came from that suspect. In this slide, I wanted to bring your attention to the characterization and the identification of the victims and the human remains from the mass disaster by Southern blotting technique. Even a certain degree of DNA degradation was likely to be neglected by the analysis of small polymorphic regions known as STRs or short tandem repeats, which are widely distributed all around the genome. The availability of this new analytical approach contributes to the development of the strategies that permit the identification of decomposed corpses and the fragmentary human remains, often the byproducts of mass disasters. This is possible by the comparison of their genetic profiles with their putative relatives. In India, this technique was introduced by late Dr. Lalji Singh Tandon, the father of DNA fingerprinting in India. The legacy left behind by the father of DNA fingerprinting is a testament to the scientific temperament that needs to be inculcated in every citizen, along with a passion for the social justice. He embodied the ideal scientist working in the service of humanity in this era where science and technology are increasingly being marketized. Here, we will discuss about paternity testing. As you all know, DNA, that is deoxyribonucleic acid, is the genetic material which we inherit from our mother and father. Paternity refers to fatherhood. So, the DNA paternity test uses DNA, usually taken from a cheek swab or blood, to determine whether a man is the child's biological father or not. Now the question comes, how accurate is the DNA paternity test? So I would like to tell you that DNA paternity tests are extremely accurate. A test can show with 99.9% accuracy if a man isn't a person's biological father. Then the next question which comes in our curious mind is that why do people need DNA paternity test? Establishing the paternity can be important for several reasons. It can help, like for gaining the legal rights to child support, child custody, social security, benefits and inheritance. Identifying the link to genetic conditions that can affect your long-term health. Various human genetic disease can be identified by using the accurate DNA probe from the defective gene. So, here in this slide, we are going to discuss how southern blotting will be helpful to diagnose the particular disease which is caused by genetic defects. A point mutation may destroy or create the restriction enzyme cleavage site, then the fragment site produced from the normal gene and the mutated gene will be different. And this can be easily identified by southern blotting. For example, sickle cell anemia, Huntington chorea, and Duchenne muscular dystrophy are some of the diseases which are attributable to single gene defects. In most of these, the mutation is recessive. That is, it shows its effect only when an individual inherits two defective copies of the gene, one from each parent. One goal of the modern medicine is to identify those features that carry two copies of the defective gene long before the birth, so that the mother, if she wishes, can have the pregnancy termination. In sickle cell anemia, for example, the exact nucleotide change in the mutant gene is known. The sequence GAG is changed to GTG at a specific point in the DNA strand, and that codes for the beta chain of hemoglobin. For the prenatal diagnosis, the two DNA oligonucleotides are synthesized, one corresponding to the normal gene sequence in the region of the mutation and the other corresponding to the mutant sequence. If the oligonucleotides are kept short, that is about the range of 20 nucleotides, they can be hybridized with DNA at a temperature selected so that only the perfectly matched helix will be stable. Such oligonucleotides can thus be used as label probes to distinguish between the two forms of the gene by southern blotting on DNA isolated from the fetal cells collected by amniocentesis. A fetus carry two copies of the mutant beta chain gene can be readily recognized because its DNA will hybridize only with the oligonucleotide that is complementary to the mutant DNA sequence. Now here comes the another important application of southern blotting, that is the diagnosis of disease or the identification of the infectious agents. In order to supplement the conventional methods available for the diagnosis, DNA diagnosis of different diseases have played a very important role. So, the biotechnology has provided DNA probes and monoclonal antibodies as two very effective and most sensitive tools. Actually, the diagnostic methods at DNA level have the advantages over the methods, since in these methods the genes of the parasites are examined and not the expressed product, that is protein, because it got changed in different stages of the life cycle of the parasite and also due to the environment. So, the rapid progress in recent years has been made in the development of nucleic acid based assays for the diagnosis and epidemiological surveillance of human parasite from the groups of protozoa and helminthes like Kala-Azar, sleeping sickness, human blood fugue, etc. These are some of the references which I have used to create this video. I hope the video was quite knowledgeable for you all. Thank you all for the patience listening.